Nobody else is in the Zoom, so hopefully they can show up. Okay, literally the last two slides that I want for your test material are going to be just protein synthesis. Now, we've talked about protein synthesis a little bit just in reference to what proteins are and how they fold. We didn't go into great detail. But what I want you to know is that there's a process that we have. I think I put this on the board once, where you start with DNA. And DNA contains a number of different nucleotides with different bases, A's, T's, G's, and C's. We know that A's go with T's, G's go with C's. And there's a sequence, and that DNA is double-stranded. Y'all remember this from the other day? That DNA goes, there are two strands, and the strands run opposite of each other. That becomes important because that will affect the, one side gets constructed in a continuous way, and the other one gets constructed in pieces. Now it's DNA. So all of that stuff to say, what does DNA have in it? What is the purpose of DNA? What would you say? Holds information. Okay. Information for what? Proteins. There are other things besides proteins that are information is held for, like the control of what proteins are turned on and not turned on, what genes are turned on and not turned on. There's something called switches and other things going on in there. But for our purposes, let's just say it holds information, and much of that information is for protein. Okay, and that's where protein synthesis comes in. We take this DNA and we are going to take that information out of the DNA, and it's going to get, there's a word here, I don't know if y'all remember this. Yep, transcription. I don't know, do y'all have a hard time remembering that?
Actually, I want to think about that.
Okay. Let me, a way for me that I remember this, and this is the reason for the use of this word, I'm pretty sure, is, are you familiar with the word transcribe?
Mhm.
What do you do when you transcribe something? There's more of a copying mechanism, isn't it? So if I ask you to transcribe something, I may hand you an audio. That may be one of the more common things we hear now. We look for transcriptions of like a podcast, for example. So you listen and somebody types it out. Okay. Now YouTube will automatically do some of that, but it's not always perfect. You have to go back and fix that transcription. Oh, this is a great example. Yes, should have thought about this before. So if DNA is your, let's say it's a blog, we'll go with YouTube, and you've got to transcribe it. So what is the original form of the information in your YouTube blog? It's video, but I can't, well, I can transcribe video, but is that what I'm going to transcribe?
Audio.
It's the audio, it's the spoken word, right? So I take that audio, let's actually put this example. The YouTube audio, and we're going to transcribe. Okay. So we're going to take audio and turn it into words. Okay. We're going to take it into the written word, specifically the written word, because audio is also words, right? But they're different formats. Okay. It's funny. If we think about it, words on paper don't make any sound. They're just visual, right? We know what correlates to a sound, and therefore we can pronounce them. But words on paper don't make a sound. Just like spoken words don't have a visual. We've made a system where that can happen, and they mean the same things. So DNA contains information, and it's huge. We have like 23,000-ish genes out there. Okay, which is a lot less than people thought. It's about the same as a chicken. But, you know, we'll try not to feel bad for that. There are some plants with more genes. Just so you know, the number of genes you have has nothing to do with your intelligence levels. Okay, so it doesn't make humans more or less because we have 23,000-ish. Okay, but in that information, and there's lots of genes, there's lots of pieces of DNA. Some of that DNA are not genes. So there's a lot of stuff going on. We go from DNA. So if we're going to take DNA and we're going to go through transcription, so a form of transcribing. So this is not audio to written word, are not the same formats. DNA is going to go to a different format. Do you remember what it was? It's mRNA specifically. Okay, now that's not to say that other types of RNA aren't also done this way. There's rRNA, there's tRNA, but specifically mRNA contains information for how to make proteins. Okay, and the M stands for what? Messenger. How do y'all like the review while we're also finishing this out? Now, do you see why I think that this is a great example? Okay, where I've never used this example before. Usually I use a library, and I think of a library where you can't take the book out. So what you do is you transcribe a small portion of a book that explains how to do something. Maybe you make a copy. Maybe you write it down by hand. And that's where that word transcribe really comes down. It used to be that you would take something over here and you would transcribe it. So if it's a print word, you put it in the written word. In this case, it's audio to written word. Same information, different format. Same information, different format. And mRNA is smaller than DNA. Because you don't transcribe all the DNA. What's the point? That's DNA replication, right? Why don't we take out just the gene, a small portion of DNA that we need? And that's what this one happens here. Now, that doesn't give us a protein. Okay, and that's where we're going here. As an amino acid sequence. Do you remember what this word was from here to here? It also starts with T. What's that? No, it's correct. It is translation. And I like to think of it this way because the information here gets translated into a protein. Just like you might translate a word from one language into a different language. Now, these right here are very similar languages, aren't they? They're nucleotides. So we're just transcribing this information into a slightly different form. This is like print English to written English, or audio English to written English. But here we're going to French, Russian, Greek, something different. Okay. But truthfully, it's not even that. We're going from audio language to written word product. It's even a better example. Okay, so YouTube tells me, you go to those DIY videos, you're like, okay, my toilet's clogged, what can I do? I don't know if y'all do that, I do that because I own a house now. I do this the other day, like, what are my options? So I took it, and a lot of times I read what they said as much as I listened. Okay, or maybe sometimes they even have stuff down in the comments that they'll add to it. So it's a written word, but I take that information and I translate it into a product. Okay, in this case, my product is a functioning toilet. Or maybe I'm going to build a shelf. I take that information and I put all those things together. Is my shelf the same thing as the instructions for a shelf? No. But the information is here on how to make that. The information is here on how to make this protein. Okay. And so That's why we kind of point out on the screen that DNA never leaves the nucleus. Just like our book doesn't leave the library. And while you can take your phone with you, you can't actually take YouTube out of the phone. Okay. But we can take the information. Okay. And you certainly can't take the person who's teaching you out of YouTube. Like just pull them. Wouldn't that be great? Like, I'm not going to fix it. I'm just going to pull you out of the video and you do it for me. That would be a great service. I wonder if we could figure that out. But that's not how it works. So what we do is they take these videos and they give us information. And sometimes in different forms that tells us how to do something, how to make something, how to whatever. Okay. Now sometimes it's not a product. It could be a process that DNA does, which a lot of times does involve a product, but it's not just, oh, I want to build a shelf. Sometimes it's, oh, I don't want to just build a protein, but I'm going to build an enzyme. And we talked a little bit about enzymes. Enzymes are going to mediate reactions so that they go faster. So a lot of these information that's over here will affect the process of something that happens in your body by making a product. Does that help to remember transcription, translation? Because that's really where those words come from. It's the idea of transcribing from a form into a form that's a little bit different. Written word versus, sorry, the printed word versus the handwritten word, for example. Same information, same language, same information all encoded there. And then he translated it into something very different, like another language or product. Okay. So DNA requires a decoder, and that's this process that takes place in transcription. And I'm not making y'all know the details of that. Okay, but transcription is that process that decodes DNA. Okay, there are other enzymes that go in and the information, and it produces this. So this is what I would love you to know, this process. Do y'all remember what I called it the other day? Central dogma. So if you ever hear this term, the central dogma of biology, we're talking this. DNA transcribed to mRNA, translated to protein. Now I'm going to throw in one little step that we've already talked about, and it's kind of right here. So after you make this amino acid, do you have a protein? What do you have to do? The structures. So this amino acid is what comes out of the translation. Then, before we get something functional, we have to go through the folding process. So we go amino acid sequence. We've got alpha helix, beta sheet. We've got our globular structure, sometimes our barrels. And here we've got, well, a lot of tertiary structures put together. Okay, so that means like for this to happen, we have to go through this 3, 4, 5, 6, whatever, however many times. Like hemoglobin, we go through this 4 times, get 4 of these, fold them 4 times, and then cook them all together. I think I just saw your processing taking place. Your eyes were like, okay. Come on. And this kind of just is a diagram that shows those kind of same steps. So you've got your DNA, go away. Oh, no, not that. This go away. It's covering. So you've got your DNA, your RNA is produced, it comes out. We go into step 3 of transcription. And there's a lot more detail here. I'm not going to make you know that. I don't think you need to. I think for an everyday kind of understanding of biology, this is important. So while your cells are in interphase, it's doing this. Not during mitosis, not during that growth phase of, I shouldn't use the word growth, not in that dividing stage. I shouldn't use growth because there are growth stages of interphase. But while the cell is actively dividing during mitosis, it's not doing this. The DNA is not available. The DNA is condensed into chromosomes. Do you remember when we talked about how we unwound the DNA during lab? We actually broke down proteins so that we could string it out and play with it. Well, in the cell during this time, a lot of this DNA is much more unwound so that it's accessible for proteins to get in and touch it. But during DNA replication, we replicate more, and then later in mitosis, we'll wind it up really tight into chromosomes so that we can move them when we separate the cell into two more easily. We good there? Okay, so that was to try to review like a little bit of cell division. Have any of y'all like looked at the little videos? They've been useful. You might do that before Friday. Okay. Yes. So I'm going to work my way backwards, I think, in terms of review. Do y'all have any questions with our 3A? So cell division, membrane transport. What are we focusing on membrane transport? Passive versus active transport. There are a few processes. So what's useful here is to just kind of remember this. Let me go back. You've got filtration. You've got facilitated diffusion. Well, we've also got this plain diffusion, which isn't really, well, it's included here with osmosis. Okay, so there are multiple types of the passive. What's the biggest difference between the passive and the active though? Active transport uses energy, passive doesn't. Why do we have to use energy as active transport? Okay for me to erase this. Remember our water example? If I spill water on this table and it falls onto the floor, it requires energy for me to do what? Not just to pick it up, it's a little bit more than that. It wasn't just to pick it up. We also talked about putting it back inside of its container. And what am I doing? If that water gets poured out, it spreads out, right? It's going to lower and lower and lower amounts of concentration. The more it spreads out, the less it gets. So active transport is when we try to concentrate it back into a smaller location. Okay? Because what do things usually do? Entropy, just in general, does this. Everything goes from high concentration to low concentration. Okay, that's why passive is passive, because just the way the physics work, everything wants to spread out from higher to lower concentration. They want to cross from higher concentration of water outside the cell to inside the cell sometimes. Okay, but if I need to get water out of the cell and it's lower concentration than the outside of the cell, how am I going to get water out? So imagine you've got a cell. Okay, and let's just, I'm going to make X's. This is going to represent water molecules. Okay. What has more water, outside or inside? Outside. Okay, so where's the highest concentration of water? Outside. Where's the lowest concentration of water? Normal osmosis, which is just diffusion for water. Passive transport, where is the water going to move? It's going to move from here in to try to increase this until it's balanced. What's the word for balance? Homeostasis. We're going to try to create a homeostasis on either side of this membrane. So water molecules will move in. But let's say for some reason the cell wants to maintain a lower concentration here than there. So as these are trying to come in, what's the cell going to do? It wants to keep it, because that happens with some molecules, not just water, sodium, potassium, other things that we're going to do. If we want to get stuff out, but it's a higher concentration outside, you have to use energy. What's the word we not want to use? It's active transport. But what's the verb? Almost. So there are pumps. Okay, we have pumps that move things in and out of cells. And this, anytime you think of pump, the reason I like to use this word is if you pump water out of the ground, do you ever use those old timey or seen those old timey like water pumps? Okay, water comes up, but does it cost energy? Your energy, right, to actually pump it. If you have a basement that gets water in it, some people have these pumps that pump water out. Okay, it costs electrical energy. To pump anything out is going to require energy. So when I think about transport, I kind of equate, this is not 100% true, but I do pump equals active transport in my head. Active transport equals pump in my head. Okay, these two things go together because it requires energy to do any kind of pumping. If you've ever pumped anything, even when you pump gas, there has to be a situation because gas doesn't just, those are in tanks underground. You've got to get them up. Okay, now sometimes you can do it with pressure and all that stuff. So maybe not always the best examples, but most pumps require some energy in some form. Okay. I actually grew up when I was young. I was pretty young when we stopped using it, but I had a well on our property. Like that's how we got water. And it required electricity to pump it and then get it where it needed to go. Okay. So energy is involved in some form, whether it's hand, electricity, or whatever. Okay. Does that help to distinguish those two? So what do you want to think? Energy and not energy. Okay. Passive is no energy transport. Active transport is requiring energy. But we also want to think about how concentrations work. Active concentrations. Anybody in social clubs? Okay, so one of the fraternities. Let me ask you this. Okay, assume your body of members is a cell. How do you get more people involved in that group?
Get that cell, get that person to bring in.
Does that require work?
Yeah.
It does. So for you to have a higher concentration of people later on in your group, it requires work to bring them in. If you don't do a lot of work, what happens to your organization a lot of times? It dies. You lose people, maybe little by little. So when an organization is passive, you go from higher concentration to lower concentration as they move away. But if you want to keep your group active and full of members, it requires work. Does that help to put it in that context? Because we see this happening not just in molecules, but in so many different situations. So passive transport, high to low. Active transport, low to high, bringing people in. Okay, so transport. Okay, there's intercellular fluid. There was some stuff we left out of the cell parts. We didn't really go by centrioles. So this microfilament, microtubules, filaments of different sorts, centrioles, those were not something we worried about. Just that you know that there is a cytoskeleton is the important thing for me. Okay. The different, I'm going to call them organelles, although it's not completely true. Peroxisomes, lysosomes, Golgi, endoplasmic reticulum. There's two forms of that. And what makes a rough ER a rough ER? Ribosomes, and ribosomes are used in what process? Do you remember? Let me show you something. So remember that there are multiple types of RNA. I've only had you focus on the mRNA, but there is an rRNA. That's where that R comes from, ribosomal. So some of that RNA is actually built into that molecule. Now here's the other thing. So what did we use RNA for before? To do the process of making proteins. Ribosomes are used to make proteins. So where are some of your proteins made in the cell? On your rough ER. Okay. Now you also have free-floating ribosomes as well that float out and about the cytoplasm, but there is a concentration of them on a portion of the ER. Okay, mitochondria, I think we're good. Do you have any questions about that kind of stuff? The cytoplasm, what's in the cell? Okay, I'm just going backwards. Plasma membrane. Are y'all fine with? I think I told y'all we're not focusing on the gap junctions, the what they call the tight junctions, desmosomes, and sorry, two different junctions there. We're more focused on that there are connections between the two. Okay, so why would you need to have connections between cells? That's what I would like you to remember. So why would you want to have cells connected in some way to another cell? Okay, how would you share information? So what would that connection look like?
So let the information be like DNA.
Well, the DNA doesn't leave a nucleus. So maybe it's not going to be in the form of DNA. In this case, a lot of times we're going to go through that central dogma, produce a protein, and send that protein out, this information. That's usually what happens. So the information is not wrong, it's just not in the form of DNA. Because we don't usually, maybe we send the RNA. I'm not sure about that. I mean, I know in the form of viruses, you can move DNA and RNA, but that's not cellular information. Probably it's going to be in the form of proteins. But still, how would you get this protein from here to here with that information? Because what surrounds the cell? There's water, but I mean, what separates the inside of the cell from outside the cell? The membrane. So this is a barrier, right? So what do you have to have? You got to have some kind of gap pore connection between those two. So sometimes there are things that allow us to go through, like the desmosomes and so on. There are ways for allowing physical objects to go through that membrane. So that's one of those connections. The other ones are like physical, remember the rivet idea? They're physically riveted together, they're held together in place. What would happen if all your rivets came apart? Think about it. If all your cells were not actually held together to each other, you would fall apart, literally fall apart. I'm very thankful that we don't have this issue. I'm picturing this in my mind, and it's creepy as I'll get at. It's like a terrible science fiction movie. Like you just disintegrate. Okay. And there's a number of ways that that happens. My goal here is just that you understand that as a multicellular organism, you physically have to have your cells connected. They don't just hang out because they enjoy hanging out with each other. There's a physical connection. Okay. Whereas with bacteria, they don't rivet themselves together because they're single-celled organisms. Okay. Do they associate with one another? Yeah, and they do some other stuff. I won't get into that to confuse you, but it's not as permanent in their case, or as tight. Okay. Are y'all okay with sugars and proteins and phospholipids and all that stuff here? Chromatin, nucleolus, cells. Y'all okay with cells in general? Okay. Now it's 8:35. What would you like to ask me, maybe from the other pieces of information? Chapter 1, chapter 2. Chapter 1 is kind of the overview of the systems. We talked about homeostasis. Chapter 2 was chemistry and all the fun that Shelby has with that. We're going to pick on her and be in the video. Hopefully she'll watch it. I think that's going to be my goal, is to see how many times I can pick on her at different points in this lab and lecture. Chapter 2? Okay. Let me pull up. I don't have myself signed in yet, but let me pull that up. I don't know why it didn't occur to me to like download the PowerPoint to this computer each time. It only took me three chapters to figure that out. Y'all ever halfway through a password and then you forget what you typed? Okay, it's good. Not the only one. And look, and I still got it wrong. I'll get to see all my classes. See all my classes. There you are. I will make sure that y'all have all the videos up from today. And sorry, that's not what I meant to click on. So I don't think I've got Monday's in yet, but I'll make sure I've got Monday's video and this one put in lecture 3. Now, Ryan, do you have anything specific that from chemistry, or just you want a general kind of go through it pretty quick?
I would just say general.
Okay.
But I feel like it's easier for me to study the chapter 1, do the system and everything.
Okay. Okay. So matter, different phases. Just for fun, can anybody tell me what the 4th phase is? Plasma. Good job. We don't see that on Earth, as far as I know. Crazy enough, that was a question in the Science Bowl a couple weeks ago that we hosted. Was dealing with plasma. Okay, we've got physical and chemical situations. Energy was a big thing, and we focused, I think, a lot. I try to kind of remind y'all about energy. Remember, can you destroy or create energy? No, it's laws of thermodynamics. We can't do that. Energy flows from high to low also. Okay, just like we've got that concentration of molecules that flow from high to low energy also happens. And that's why you go from potential energy to kinetic energy. Okay, there's a few forms of it. ATP is our molecule that we store energy in. ATP is important. If you don't have ATP, what happens? You die. Okay. So if you drown, if you get your head cut off, what actually do you die from? Lack of ATP. Okay. technically, because all of that just means that your cells run out of energy. Okay, and so those situations create the problem of not having ATP. Okay, do I want you to know oxygen, carbon, hydrogen, nitrogen? I do. Y'all of you have your quizzes back, right? Okay. Other guys, I'm looking at y'all. If y'all see this, I have your quizzes. Let me know if you want. Because y'all need to come today. See, picking on. Okay. There are a lot of other things. Why do we want to know oxygen, carbon, hydrogen, and nitrogen? Why do I make that important? Like, just to want you to know that.
Four sort of essential things that you need to do everything.
Yeah, the word we're looking for here, and you're completely right, is because we are organic, um, just a bunch of organic molecules put together as an organism. Those are the four major things involved in organic chemistry that makes up who we are. And not just humans, but every living thing. Okay. Now, are there other things involved like phosphates? We talked about phosphate. ATP has got a bunch of phosphates. But these four are the most common. So if you were to go through like and look at percentages of body mass, I mean, oxygen, 65% of our body mass is oxygen molecules. Okay. So there's a bunch of elements there. But should that be surprising? Because what makes up some of your body mass? Water. Okay, H2O. Let me ask you this. Which one has more mass, hydrogen or oxygen? I didn't. It's oxygen. Why do you say oxygen? But there's two H's. So we didn't cover this, but just for fun, we when you look at the periodic table of elements, which is the first one, hydrogen. Okay, it physically is smaller. So oxygen, on the other hand, has more protons and neutrons than hydrogen does. It physically has more mass because it has more protons and neutrons. So as you go up in molecular weight, you're actually going up in atomic mass as well. So just for fun. Okay. You don't have to necessarily know any of the others. You will see them, but don't worry about those so much. And I think we mentioned, where do you get a lot of these? These side notes here, especially. Actually, almost all of them, including your carbon and your oxygen. A lot of your oxygen, actually, and your nitrogen, and all these others. How do you get them? Do you just breathe the carbon dioxide out of the air and absorb it into your system? Yeah, it's food. Okay, food and water. It's some kind of consumable. Okay, and that includes your iron and your iodine. Okay, what's our biggest source of iodine? Probably. Huh? I can't hear what you're saying for some reason. Seafood? Well, possibly, but we actually do something to a certain supplement to our food. Iodized salt. And they do that on purpose. Okay, it affects your thyroid if you don't have enough iodine in your diet. And there are populations on Earth that still don't have enough iodine in their diet. We don't have an issue with it because, bam, we iodize our salt. But again, it's something that you intake. It's consumable. Okay, unlike plants, we don't just absorb it from the environment. We have to consume it. Okay, you got your atoms. Y'all think you're okay with protons, neutrons, electrons, the charges. Electrons bear almost no mass, not completely no mass, but very little. Y'all are okay with charges, right? Positive and negative. Are y'all okay with well, you didn't have to know this exactly, but I did want to show y'all the orbital models. Okay. What we want to know, though, is that electrons do orbit, because why is that important? The orbiting of electrons, that outer layer of electrons, is involved in making What? Bonds, okay. That's the importance here, bonds. Okay. So looking at those outer orbitals, let's see, isotopes. Isotopes, by the way, just you have different atomic masses, so you may have a different number of neutrons. Okay, we looked at this. This is the atomic number and atomic mass. You don't have to remember any of those. I'm not going to ask you. This is not chemistry class. But we just kind of briefly had some fun talking about this. Okay, molecules. So bringing two things together, like hydrogen and hydrogen together to get an H2O molecule. So you have two atoms coming together to form a molecule. Two atoms, separate atoms in and of themselves, to create an an H2 molecule. So we've got two reactants yielding a product, and it's the interaction of electrons that create a bond. Okay. We can also create a compound, and this is where you've got two or more atoms of different elements that create this. So you've got hydrogen and carbon coming together to make methane. So this is a compound. So you've got molecules, and you've got compounds. Sodium and chlorine come together, you get salts. Both sodium and chlorine are by themselves are very interesting. Salt, less so. We're about to get into different shells. You don't have to remember the different shells. Just remember that the outside electrons are the ones that interact. And when they interact, you get find ionic bonds, for example. And so what's the difference between ionic and covalent? Yeah, so we don't have any share. We have literal giving or taking. And why do those two things stay together? So it's like, oh, the sodium is going to give or take. Why do you have the sodium and the chloride staying together when an electron is literally taken away? So what does the Na and the Cl stay together? It's an ionic bond. What creates an ionic bond? What's the name? Ionic? The name's ionic, right? So what are ions? Positive or negative atoms. So the name actually tells you, because they're ionic in ionic forms, we have an example where the electron is actually transferred. So one becomes positive, one becomes negative, and because those are opposite charges, they attract one another and stay together. Okay, so that's what that ionic bond is telling us. So we get the transfer of electron fully, but one of them becomes negative, one of them becomes positive, and they stay together because of that charge. Covalent, on the other hand, you don't. Okay, they do not like to share. Well, they share, but this is the kind of sharing like, okay, my brother can have it, but I'm not going to let go. You know what I'm saying? Like, it's the toy, but you're going to hold on to it for your life. Technically, you've done what mom said, you share, but you know what I'm saying? So that's what's happening here. And how can you tell how many bonds you have? So it all depends on the amount of open electron spaces in your, like carbon has got 4. Okay, it's got 4 electrons at the outside. It always can go up to 8. So you get 1, 2, 3, 4 more that you can fill. So you get 4 bonds you can make. Okay. So that's what we looked at here. Now, covalent can be nonpolar or polar. The difference is just that nonpolar covalent bonds are equally kind of arranged so that you don't get a positive side and a negative side. Okay, so the way I think about this is you can get a really nice division this way or this way, and they're even on either side. Does that make sense? Okay, I'm not explaining that as well as I would like. But because they're not bent in any way, the molecule doesn't have something a little more on one side than the other. For example, water. Water does this, and because it does this, you get a slight positive charge, a slight negative charge, so you get a polar covalent bond. And polar covalent bonds can also interact with one another. This leads to hydrogen bonds between water molecules, because water will arrange itself like this. So if this is, so you'll get another water molecule like that. Because you've got a positive side, a negative side, a positive side, a negative side, a positive side, a negative side. Okay, and this is hydrogen bonds. And it's not just between water, but you also get this between DNA strands. We see parts of our folding of the protein. So part 2, the alpha helix and the beta sheet are hydrogen bonds holding those together. Okay. If you've ever seen a little water bug running across the top of water, have you ever seen that? It's cool. They do it because water actually has some tension because of these bonds, and they don't weigh so much that they break that tension and they just scurry across the top of water. So cool. Okay. Reactions, remember we didn't need to know synthesis, decomposition, all that stuff, just that reactions. We've got reactants yielding products. The arrow is the yield sign. There are different ways of doing that. Okay, let's go back water. Why is water important for life? It's got a high heat capacity, it's polar, it's chemically reactive, it allows for cushioning, dissolving of things like salts. They disassociate in water very easily. You suck the water back out, dehydrate the water back out, those bonds of the Na and the Ca will go back together. We talked about acids and bases. Are y'all okay with that? Okay, so again, what's an acid? The acid is going to be somewhere from 0 to 7. Bases are 7. Well, actually, 7 is not, it's neutral. But from 7 to 14 are bases. Okay, so acid. It's all about the number of OHs or H's that you've got. Okay, you can neutralize. We talked about. Okay, your big major organic compounds, sometimes called the macromolecules. We've got carbohydrates. So make sure that you know your carbohydrates, lipids, especially phospholipid correlation there, important. Proteins, which we've just had some review of. And nucleic acids. Okay, which ATP is involved is one. So, okay, that was as quick as I could do it. Hopefully that's okay. I am over by 4 minutes. I apologize. I will see y'all Friday, and we'll take a test.